Hi, my name is Emma Dickinson, and like many of you, I like to spend a lot of my time outside. However, this does expose us to unnecessary risks of ticks, and with the help of my research advisor, Matt Persons, we are studying deer tick attachment to different fabric types. Like many people are already aware of, ticks are vectors for many diseases, and they pose significant health risks to humans and animals. A lot of people are familiar with Lyme disease, but there are also other diseases like anaplasmosis and babesiosis, which are extremely relevant in our area. A tick's host-seeking behavior is called questing, which I have a picture of here, and generally involves sitting at the top of vegetation and waiting for a suitable host to pass by. Ticks grab onto our clothing, and that's how they get to us and use us as a host. Before we start talking a little bit more about what I'm going to do, we need to hammer down a few points about the tick life cycle. First of all, the larval ticks, which is the type that I'm working with, they cannot have Lyme disease. They require a blood meal in order to get it, and as you can see in this little life cycle, from going from egg to larva, there's no blood meal. So they cannot acquire any sort of Lyme disease because they haven't been fed yet, and the bacteria cannot be transmitted from adult to their offspring. Ticks are also prone to desiccation, meaning that they dry out, so their housing has to be very particular in order to keep them alive in the lab. Also, ticks find their host with a Haller's organ, so this is found on their front two legs, and that is why they wave them around when they're questing. This allows them to detect carbon dioxide, humidity, heat, odors, and pheromones. And right now, the technology that we have to estimate tick population density and distribution are drag cloths. This is a big piece of fabric that gets dragged behind a researcher on a string, and questing ticks grab onto it. And then you can count how many there are and use the area covered to determine your population density of ticks. So these are some important background notes just before we get into some of the actual research I'd like to do. So some things that we do know. We do know that tick nymphs of Ixodes scapularis, which is the deer tick that I'm studying, 62% of the ticks in this study attached to light clothing versus 38%, which attached to dark clothing. More of the nymphs were attaching to the light than the dark. This is pretty astounding because we are normally told to wear light colored clothing so that you can see the tick when it's on you and remove it to prevent disease. But this 2004 study kind of turns that around, suggesting that clothing color does actually matter. Then in 2021, there was a study that determined that the material that you are using for drag cloths also matters in your tick acquisition rates. So they tested canvas, corduroy, and flannel drag cloths and found that the number of larval ticks collected was greatest with the flannel and lowest with the canvas. But canvas, corduroy, and flannel are not usually materials that are worn in the field. So, despite this important information about tick attachment, there are no studies that have tested the performance of the types of materials that we wear every day. If there's a material that ticks grab onto less, it might influence our standard dress codes for field work. Clothing is the first barrier against ticks, and we should be testing the types of fabrics that we're most likely to wear when we encounter them. So, what I want to know is, do ticks adhere to fabric types at different rates? And if so, which fabrics perform the best? So, this is how I'm going to do it. I'm going to take a damp soil substrate, add 10 ticks in, blow on the ticks to stimulate questing. They have the Haller's organ to detect the carbon dioxide from me, and they'll think, oh, there's a host, and start their questing behavior. Then I'll lay the fabric swatch on top of the ticks and wait for a couple seconds and wait for them to attach, and then record the number of recovered ticks. These are the fabric types that I'm planning on working with. So these are all white. They do not look white, but I assure you they are. Shout out to Shadows. They're all white because of that first study that we looked at in 2004, which showed that the color does show it shows a difference. I don't want ticks to be attaching to different fabric textures and types and materials based on the color. So I've standardized all of that here. And here is where color's out of the question. These are samples of actual clothing that I was able to locate at a thrift store of the exact same materials as on the previous slide. Those are the ones shown with the green background. So these are actual clothing materials. They've all been washed in the same detergent as the first ones. And I'd like to compare across the color as well as between the fabric types. So what I've done so far is I've acquired the ticks. That was a huge step. I have about 9,000 ticks in the lab right now. And I've also set up humidity chambers with potassium sulfate in order to keep the humidity at about 98% so that they don't dry out. I've also prepared my fabrics, as in I have them and I washed them. And I've also developed an extensive lab safety protocol because I am not interested in releasing a bunch of ticks in our lab. So they're stored in these little vials here with a hole punch lid type thing going on, and they have a 250 micron nylon mesh mashed in there to keep the ticks in and to let the humidity exchange freely. Then they go in a airtight Tupperware container where the humidity is maintained with the potassium sulfate. Then, when I work with them, I wear a white lab coat, white gloves, and work in a contained unit that has a petroleum jelly ring because ticks can't cross petroleum jelly. It's just too thick. And that red thing I'm holding is a paintbrush because it's really hard to move them. They're very small, so I use a paintbrush for that. The next steps are to dry all the fabrics that I have. Here's a picture of them drying outside on the loading dock. And I also need to dry all of the soil substrate that I'm going to be using so that I can standardize everything. Then I need to determine the soil and water ratio to create my damp substrate so that that's also standardized. Then I'll run all the experiments with all of my fabric types, and that will take a really long time. Then I get to analyze my data. Hopefully I'll find something interesting. And then I get to dispose of all of my ticks, which should be cathartic. So I want to identify fabrics that recover the lowest proportion of ticks, but I also want to identify the fabrics that could recover the highest proportion of ticks. So for the highest proportion of ticks, that could be applied to drag cloths. Materials with high capture efficacy could be used for population sampling in order to determine density and distribution across Eastern America or Pennsylvania or just around here. And then the materials that don't collect that many ticks could be ideal clothing options for going out into the field in areas where you may encounter ticks to reduce our exposure to something that could give us a disease that is not so fun. I've had several tick-borne illnesses, and our clothing really is the first line of defense that we have. And if we can find a material that does not allow ticks to attach to it as easily as others, it may influence the clothing that we are choosing to wear. So these are all the places I got all my beautiful graphics. Shout out to SlidesGo for making my slide design easy. And I'm super excited to see what comes next because I really believe that tick research is something that is really overdue. We need more updated information, and I'm really excited to provide some of that. Thank you.